This is the story of how a single drop of something bitter on your tongue can rewire your nervous system in under 5 minutes, and how modern science is only now catching up to what ancient physicians understood 3,500 years ago. Bitter receptors were thought to exist only to prevent poisoning. They have now been found across your entire body, and what they trigger there changes everything. A drop of bitter liquid lands on the tongue. Within seconds, molecules lock onto receptors that have waited for this moment since the dawn of vertebrate evolution. These are the T2Rs, once dismissed as primitive poison alarms, now recognized as a network that reaches far beyond taste. The cascade begins. Calcium ions surge inside taste cells, sparking an electrical burst that races along cranial nerves to the brainstem's solitary nucleus. The signal does not stop at perception. It triggers a handshake with the autonomic core, the vagus nerve. The timeline is precise. In the first 30 seconds, the body flickers into alert. Heart rate rises, vessels tighten, a sympathetic jolt. Then, as the bitter taste lingers, the balance tilts. By the 3-minute mark, the parasympathetic system takes command. Heart rate variability, HRV, climbs, signaling vagal dominance. Blood pressure drops. The body shifts from vigilance to restoration, all within a 5-minute window. These changes are not subtle. Human experiments show RMSSD, a key HRV metric, rising by over 20% within 2 minutes of a bitter sip. In animal studies, vagal nerve firing peaks at 30 seconds and holds for several minutes. The effect is reproducible, measurable, and remarkably rapid. This is not fringe wellness, not a biohacker's trick. It is a reflex, hardwired, ancient, and visible in real-time physiological data. The bitter reflex transforms a fleeting taste into a full-body event, proving that the tongue is not just a gatekeeper, but a trigger for systemic change. 5 minutes. That is all it takes for a drop of bitter to tip the scales of the nervous system. 3.5 millennia before the term receptor entered science, healers were already reaching for bitter roots to treat the gut and calm the body. The Ebers Papyrus, written around 1550 BCE in ancient Egypt, lists remedies for digestive distress that rely on bitter botanicals, gentian among them. The word for bitter appears beside plant names, signaling a deliberate choice, not chance. In these earliest medical texts, bitterness was more than a warning, it was a tool. Centuries later, Hippocrates, the father of Western medicine, classified amara, bitters, as agents that could stoke digestive fire and clear excess from the system. His Greek texts described the taste as both a sensation and a category of medicine. By the 1st century, Dioscorides de Materia Medica cataloged gentian and wormwood as standard bitter decoctions for appetite and liver complaints. These were not minor footnotes, they became pillars of the pharmacopoeia, their use spreading from the Mediterranean to medieval Europe, where gentian became a fixture in theriac, the era's universal antidote. Detection was precise. Gentian's bitterness can be sensed at 1 part in 50,000, about 20 micromolar, making it one of the most potent, naturally occurring bitters known. Ancient practitioners did not know the molecule's name, but they knew its power. For thousands of years, the same plant cycled through prescriptions and rituals, their bitterness trusted to shift the body's state. This empirical tradition, written in papyrus and parchment, built the foundation for a question modern science would only begin to answer. Why do bitters, tasted on the tongue, ripple through the body so quickly and so thoroughly? At the turn of the 21st century, the search for the molecular roots of bitter taste entered a new phase. In 2000, researchers isolated the first human bitter receptor gene from a fetal tongue, cDNA library, using degenerate PCR primers designed from conserved motifs found in G protein-coupled receptors. This became the prototype for a family now known as TAS2. Within 2 years, the field exploded. A systematic cloning pipeline identified 11 more TAS2R genes and 4 related pseudogenes. Each new sequence was cloned, expressed in HEK293 cells, and tested for calcium responses to classic bitter compounds like quinine and denatonium. The evidence was clear. These were functional receptors, each tuned to a spectrum of bitter chemicals. The standard workflow of cDNA extraction, heterologous expression, and calcium imaging established a model for deorphaning the entire receptor family, a process that continues today. The story did not end at the tongue. In 2002, a paper in the Proceedings of the National Academy of Sciences reported TAS2R transcripts in human enteroendocrine L cells. When these gut cells encountered bitter agonists, they released GLP-1, a hormone essential for glucose regulation and appetite control. The connection was direct. Bitter sensing in the guti could trigger metabolic signaling, not just taste perception. Quantitative RT-PCR and ex vivo perfusion of human and mouse intestine confirmed that the same receptors ancient healers exploited for digestive effects were now mapped at the molecular level. The gut's ability to detect bitters and release hormones pointed to a new target for metabolic disease. What began as folklore, bitters for digestion, became a pathway for drug discovery. By the early 2000s, the bitter reflex had moved from empirical tradition to molecular reality. The cloning of TAS2RS and the discovery of their gut signaling roles transformed bitter taste from a primitive warning system into a druggable network. The groundwork was set for the next revelation. Bitter receptors were not confined to the tongue or gut, but were distributed throughout the body, waiting to be understood. In 2010, Stephen Liggett and his team set out to answer a simple question. What happens when bitter compounds meet the lungs? The assumption, grounded in decades of textbook physiology, was that any activation of airway taste receptors would trigger constriction, an evolutionary defense against inhaled toxins. But the results overturned everything. Using strips of freshly isolated human airway tissue, Liggett's group applied denatonium, a molecule notorious for its intense bitterness. Instead of tightening, the airways relaxed, fast and far beyond expectation. At peak, denatonium delivered 85% relaxation of pre-contracted airway muscle. For comparison, isoproterenol, the gold standard beta agonist used in asthma inhalers, managed just 28% under the same conditions, 3 times the effect in a matter of minutes. Liggett later recalled that they first thought the bitter taste receptors in the lungs would prompt a fight or flight response, but that's not what they found. The effect wasn't subtle. It was so robust that pharmacological blockers could erase it entirely, proving the response was driven by the bitter receptors themselves. T2RS, embedded directly in the airway smooth muscle. This was the first hard evidence that the bitter reflex could reach beyond the tongue and gut, directly altering airway physiology in a way no one had predicted. For the millions living with asthma and chronic lung disease, the implications were immediate. A new therapeutic target, hiding in plain sight. The body's ancient warning system had become a tool for powerful, rapid bronchodilation. The story of the bitter reflex now stretched from ancient roots to a modern laboratory, with the stakes suddenly much higher. The diversity of bitter taste receptors is a story written across hundreds of millions of years. Early vertebrates faced a world of untested plants and toxins. By around 200 million years ago, the first T2R genes had already begun to multiply, each new copy tuned to a different molecular threat. Today, the human genome carries 33 TAS2R loci, 25 of them fully functional, the rest silenced by mutations. These genes are scattered mosaics, not a single cluster, and their protein products form a family of 7 transmembrane receptors. With a paradoxical design, the intracellular regions are nearly identical, but the extracellular binding pockets are wildly variable. This variability is no accident. It means a single species can sense thousands of bitter chemicals, yet each receptor is selective enough to avoid constant false alarms. For decades, this complexity was a barrier. Drug designers struggled to target one T2R subtype without hitting others. But the landscape shifted in 2025 when AlphaFold 3 produced high-confidence structural models for all 25 functional human T2Rs. These models revealed the fine details, pocket volume, charge, and hydrophobic grooves that define what each receptor can bind. TAS2R14 emerged as a central player, showing the highest expression in the human gastrointestinal tract, and a binding site broad enough to recognize both plant alkaloids and synthetic drugs. The promise is clear. With these digital blueprints, chemists can now aim for subtype selective agonists, potentially unlocking therapies that act on the gut, lungs, or immune cells without off-target effects. The challenge remains steep. Each receptor's mosaic expression and overlapping specificity demand precision, not just brute force. But for the first time, the ancient logic of bitter detection is being mapped at atomic resolution, turning a defensive reflex into a platform for rational drug design. Clinical evidence now links the bitter reflex to more than digestion. In Japan, a 12-week randomized controlled trial gave older adults iso-alpha acids from hops. Those who took the bitter compounds daily saw clear gains in memory and attention, with stress markers falling and cognitive scores rising by several points. These improvements matched increases in vagal tone, suggesting a direct connection between bitter-triggered vagus activity and brain function. In animal studies, researchers at the University of Colorado paired vagus nerve stimulation with motor learning tasks in mice. Mice receiving brief, timed pulses of stimulation learned new skills almost twice as fast as controls. This points to the vagus nerve as a powerful lever for brain adaptation, whether activated by electrical means or taste pathways. Early research now asks if bitter compounds could offer a non-invasive route to similar effects in humans. Translating these findings is complex. Bitter receptors bind dozens of unrelated molecules, making drug design unpredictable and raising the risk of off-target effects. Regulatory barriers are high. Supplements must meet generally recognized as safe standards, but any therapeutic claim triggers full drug approval, demanding years of safety and efficacy data. No agonist selective for T2R receptors has reached late-stage trials. The promise is real, but so are the hurdles. 5 minutes. That's all it takes for a drop of bitter to shift your autonomic state, measurably, reproducibly, and far beyond the tongue. As AI decoding accelerates and receptor maps fill in, what once was ancient empiricism is now molecular design. Not fringe wellness, not a hack. This is biology, rediscovered,